Welcome to Let's Get to the Marks. We're going through the Biology GCSE Combined Science Trilogy paper for 2021. Make sure you subscribe, hit the like, leave any comments, questions, anything you want to ask. All right, let's get into it. I recommend you pausing the video at the start of each question before getting into it. All right, let's go. Question 1, Figure 1 shows the glands in the human body. We've got A, B, C, and D, 4 different glands, and 1.1 says which organ system includes the glands shown in figure 1. What glands do we have? We've got the pituitary, the thyroid, the adrenal gland, and the pancreas. So which organ system contains all 4 of these glands? Well, what do they all release? All of these glands release hormones. The adrenal gland releases adrenaline, the thyroid gland thyroxine. So you might want to put the hormonal system, but that'd be the wrong answer. The correct answer is the endocrine system. These are endocrine glands. Which gland produces insulin? Well, that's going to be D. You should remember that the pancreas produces insulin and glucagon. They help with sugar control in the blood. So, which gland also produces hormones that stimulate other glands to promote or produce hormones? Well, that's going to be A here. That's the pituitary gland, and it stimulates the thyroid gland and it acts on the ovaries, which aren't shown in this diagram. So, the answer here is A, which is the pituitary gland. It's sometimes known as the master gland, and the other glands are like slaves or servants, as it basically tells them what to do by acting on them. How do hormones travel from one gland to another gland? So, how does the pituitary gland communicate with these other glands? And of course, hormones are chemicals that dissolve in the blood, so they travel in the blood. Right, the next question 1.5. Name 2 glands involved in human reproduction. Do not refer to the glands in figure 1. So, we can't really refer to the pituitary gland, which produces FSH and LH, which is part of the menstrual cycle. What glands are involved? Well, you can think here we've got the ovaries circled in blue. The ovaries produce progesterone and estrogen. And the testes, what do? Well, they produce testosterone and sperm. 1.6 Ovulation test kits can help women know when they are most fertile. Ovulation test kits detect the increase in the hormone that stimulates ovulation. Ovulation, that's the release of the egg. Which hormone is detected? So they're basically asking you which hormone causes the egg to be released. Well, FSH is follicle stimulating hormone. That's the first one. And that stimulates the development of the egg. Now, estrogen, estrogen doesn't maintain the lining. Estrogen actually develops the lining. Maintaining the lining of the uterus is progesterone. Estrogen develops the lining of the uterus. So that just leaves us with LH, luteinizing hormone. And LH causes ovulation, which is the release of an egg from the ovaries once a month. Okay, time for a 6 marker. A new contraceptive drug for men is being tested. The drug is given in one injection. It stops sperm being able to fertilize eggs. It's effective for up to 13 years. Evaluate the use of the new drug compared with existing contraceptive methods. So here I've gone ahead and slammed all the answers onto the page. So first thing, it's not permanent, which is just like condoms, same as the estrogen pill, or not having sex, which is called abstinence. Compare that to surgical sterilization, where you can't change your mind. That's where you get your tubes cut. Also, no operation, so there's a lower risk of infection. It relies on men, which is just like condoms rely on men, which is unusual as most of the methods of contraceptions rely on women. Now, there's lots of disadvantages. It doesn't give you protection from STD, sexually transmitted diseases, which you get with condoms. It doesn't last as long as permanent sterilization. But also, there's unknown side effects. This is a clinical trial. It tells us in the stem of the question there it's a trial. And when you get an injection, there is a greater risk of an infection than you would if you took an estrogen pill or if you used a condom. Right, here we got question 2. It says figure 2 shows the money spent on conserving biodiversity in the UK of the government. On the left-hand axis, we have money spent in millions of pounds, and then we've got the year at the bottom on the x-axis, and it shows from 2005 to 2017. The question says, describe the trends in the money spent on conserving biodiversity from 2005 to 2017. Well, look, from 2005 to about 2008 or 9, it increases. So we could say, well, the money spent increases at first, and then look what happens. It decreases from about 2008, 2009 to 2011. So really your 2 marks here is for describing those 2 parts to the pattern. First of all, it goes up and then it slowly goes down. Once again, make sure you hit the pause button before each question and you have a go. So for here, you got 2 marks, and really it's just for saying that it goes up and then it goes down. Best to use the data from the figure, as that's what it asks. Okay, now we've got a maths question, 2.2. Calculate the percentage decrease in the money spent on conserving biodiversity between 2013 and 2014. They've given us the equation here, and they want the answer to 2 significant figures. So all we've got to do is pull out the information. So first thing, let's underline the year, 2013 to 2017. Draw a line across from 2013. That's 690 million. Okay. Now let's go to 2017. Let's draw a line across to the y-axis. And this is going to work out to be about 460 million. So we now know what the decrease was. It was from 690 to 460. How much did it decrease? Well, we need to do 690 subtract 460. And if we do that, we're going to get 230 million. So 690, take away 460, which is 230 million. And then we divide that by the money spent in 2013. And that was 690 million, the original, times by 100. And we're going to get our percentage difference, our percentage decrease. And the answer is going to be 33.33%. But check, this wanted it to 2 significant figures. So we're just going to do 33. We would have gone to 34 if it had been 33.5. But as it's less than 5, we round it down. So it's just going to stay as 33%. Okay, coolio. Let's move on. Let's go to 2.2. Conservation of peat bogs can help maintain biodiversity. Give 2 uses of peat taken from peat bogs. Well, one is fertilizer, and the second sect is that peat is combusted as a slow-burning fuel. Okay, let's move on. So your 2 key answers here are fertilizer and fuel. Let's move on. 2.4. Describe 2 ways, zooming in here, to increase biodiversity in the UK. Okay, there's loads of ways. Don't refer to the increase the amount of money spent or talk about peat. Right, well, we can do something called rewilding, which is taking things from zoos or from seed banks and re-putting them out into the wild. We can plant more trees to create more habitats. We can ban hunting. There's a whole heap of things we can do here to actually increase the biodiversity. We can get farmers to grow more hedges around their fields. We can reduce pollution. We can introduce breeding programs in zoos. There's a whole heap. You can pause the video and check those answers out. All right, let's move on. I'm looking to get through this video nice and quick, people. So, number 3, a fossil was found in rocks. The rocks were formed from mud. The fossil is of the fungus Aurospharia. I don't even know how to pronounce that. Giraldae. Anyway, what is the genus of the fungus? This is known as the binomial naming system, and always the species is the one in lowercase, the second word, but the first word is always the genus, the one that begins with a capital. So it's Aurospharia is the name of the fungus, the first word. Now, you know you're dealing with a binomial system. It will all be in italics, so the words will look a bit freaky. The next question, why was mud important for the formation of a fossil? Well, the mud surrounding the fossil prevents oxygen getting to the organism that's supposed to be decaying. This means the bacteria can't do aerobic respiration and they can't fully decay that organism. So that actually prevents it fully decaying and helps form the fossil. There we go. Let's look at 3.3. The estimated age of the fossil was in a range from 8.9 times 10 to the 6 years old to 1.1 times 10 to the 9 years old. Actually, that's 8.9 times 10 to the 8. My eyesight's terrible. To 1.1 times 10 to the 9 years old. So calculate the range of the estimated age. We need to take the bigger number and subtract the smaller number. So our bigger number is 1.1 times 10 to the 9, basically 1.1 with 9 zeros. And our smaller number is 8.9 times 10 to the 8, basically 8.9 with 8 zeros after it. So whack that all into the calculator, and what do we get? So the answer is 2.1 times 10 to the power of 8. So 3.4, humans did not exist when the fungus was alive. So just one other reason why an accurate estimation of this species of fungus or when this species of fungus existed is not known. Okay, so there were no humans around. They've said, forget about that. What's another reason? Well, the current technology we have now for dating fossils is not perfectly accurate and may not yet be the best. Also, lots of fossils have been destroyed by geological activity. You should all know this. We've only got about 1% or 0.1% representation of all the living things through fossils. Most things do not become fossils. Maybe we haven't even found the earliest fossils of this fungus yet, and we still haven't dug in areas or found early versions of the fossil. Okay, that'll get you your 3 marks. Now let's move on. So this is part of the three-domain system of classification created by Carl Woese, and they're not archaea and they're not bacteria. They're actually eukaryotes. There's a reason for that. You don't need to put the reason, but it's because they got a nucleus, they got mitochondria. Basically, they have double membrane-bound organelles, things like mitochondria. So they're eukaryotes. That's the answer you need. Let's zing. Okay, looking at 3.6, which two characteristics are features of organisms in the domain archaea? Well, Carl Woolsey came up with the three-domain system, which splits into eukaryote, bacteria, and archaea. So, which one of these fits into the three-domain system? Well, let's have a little look. Can survive in extreme environments, can only survive in light. Right. The archaea bacteria are an ancient type of bacteria that survive. They're called thermophilic bacteria, some of them, that can survive in volcanoes. So they're extremophiles. Also, just like all bacteria, the archaea bacteria have chromosomal DNA or a loop of free floating DNA. Moving on, 3.7, Carl Linnaeus lived in the 1700s. Carl Linnaeus classified things into groups depending on their appearance. Give three types of evidence that are now used to classify things. Do not refer to appearance in your answer. So, three ways we can compare how similar things are and group them. We can use light microscopes to see if there's a nucleus or cell wall in both organisms. We can use the electron microscope to see smaller things like mitochondria or ribosomes, and we can compare those when grouping things. We can also look at the DNA base sequence and see how similar the DNA is between two organisms, so between humans and chimpanzees, and see how closely related they are. We can look at chemicals floating in the cytoplasm, things like proteins, and we can look at the amino acid sequence of the proteins. We call this chemical analysis. We can also look at the processes that animals do. For instance, plants do photosynthesis, but humans or animals don't do photosynthesis. So we can look at the types of processes like aerobic respiration. Some organisms do aerobic respiration, others only do anaerobic. So these are four different ways we can look at comparing things without looking at the appearance. Figure 3 shows one species of bird on a bird feeder. Birds use their beaks to reach nuts inside the bird feeder. Figure 4 shows the beak length of this species of bird in the UK. The species often visits bird feeders. We can see there's the mean beak length on the y-axis or the left-hand side, and then underneath on the x-axis is the time. What's happening to that line over time? Well, we can see it's going up. This means the bird's beak length is increasing, and this question wants us to calculate the beak length between 1984 and 2000. And whenever they ask you for the rate of change of beat length, whenever they ask for rate, you've got to divide something by time. In this case, to find the rate, we're going to divide the length of the beak length by time. So beautifully for us, they put this black line out here. Now they wanted us to do 1984 to 2000. So zing up to 2000, draw a line across to the y-axis, circle. We're looking at the beak length, maximum of our range of 13, 60. Now, it's quite difficult to find 1984 at the bottom here. So what we're going to have to do is I can find along the bottom there are 25 squares between 1980 and 1990. That's 10 years. 2.5 squares per year, 2.5 times 4. If I move along, 1984 falls at 1355. So I do 1360 minus 1355. That equals 0.05 millimeters. I now need to divide that by the total number of years. And that's 2000 minus. Sorry, a bit of audio trouble there. Yeah, that's 2000 minus 1984 is 16 years. So we do 0.05 millimeters divided by 16 years, and we're going to get 3.125 times 10 to the minus 3, or 0.003125 millimeters. And that's your rate of change in millimeters per year. All right, let's move on. 4.2. Explain the process of evolution that could cause the trend in figure 4. Well, we're looking at the trend in figure 4 is that the bird's beaks increased in length. So that's the trend, that the bird's beaks increased in length. They got longer over time. So what we have to ask ourselves is, what is this process of evolution? And you should know that the process or mechanism behind evolution is called natural selection. So this is just a standard 6 marks answer we go for any natural selection question. So a random mutation equals a change in a gene, which leads to a new allele or gene for longer beaks. Birds with longer beaks, more likely to get more food, more energy. You can just roll with whatever it is in the question here, which means they're more likely to survive. Now, that's the important bit. The change in the gene makes them more likely to survive. Therefore, they're more likely to reach breeding age, mate, and reproduce. Once they reproduce, they have children, which we call offspring, and they pass on that gene, that allele for the longer beak, to their offspring. If this happens over many, many generations, the number of genes for long beak in the population will increase. And this process is called natural selection. Now I can spot a word I missed out near the top. We could have said you get a random mutation which leads to variation as well as an extra marking point. Now, I just want to tell you that the question could have been, why do leopards have spots? And it's the same answer. It doesn't matter what they ask you. As soon as you know it's about evolution, you say, random mutation gave a leopard spots, gave a change in a gene, which caused some leopards to have spots. This made them more likely to survive. You can make up why, better camouflage or whatever. That means they're more likely to reproduce, have sex, and have children, more likely to pass that beneficial allele or gene onto their offspring, their children. And this happens over many generations. And so it's about any feature they come up with. If you get the clue it's about natural selection, the answer's the same every time. 4.3. Birds of this species live for about 3 years. That's a short time. And they produce up to 24 eggs a year. That's a lot. Explain why evolution is easier to study in this species of bird than in humans. Well, they produce lots of offspring, 1 mark, which have shorter lifespans, another mark. This means it's easier to study natural selection. Natural selection happens faster, or the genetic changes in natural selection happen faster. As a side note, geneticists often study fruit flies because they have even more offspring and have even shorter generation. Just a few days is their lifespan. Right, let's move on. Describe evidence that would allow two different or individual birds to show they are of the same species. Right, well, if they're of the same species, you can reproduce and produce fertile offspring. So we should be able to get these two birds, get them to reproduce, and then get their children and get them to reproduce. And that would mean they are of the same species. So they can reproduce and have fertile offspring. Now, some other evidence, we can study their DNA and see if the DNA between the two birds is similar. So we can do DNA analysis. Then we can do chemical analysis and see if the chemicals found in them, it's called molecular evidence. We can see if that is similar as well. And of course, we can also use old Carl Linnaeus's methods, which was to see if the appearance of the birds, the phenotype, was similar as well. So here you're scoring marks for the DNA sequence is similar, the appearance is similar, and can they produce fertile offspring? Right, let's zing on. Question 5. Caffeine is a drug that decreases reaction time. A group of 16 students investigate the effect of caffeine on reaction time. The students were all 15-year-old girls, so they kept that control variable the same. The group was divided into 8 pairs of students. This is the method used. Student A starts 2 stopwatches at the same time, boom, boom, then hands one of the stopwatches to student B. Student A says stop at the same time as stopping her stopwatch. Student B stops her stopwatch as quickly as possible after student A says stop. The difference in the time between the 2 stopwatches is recorded. This is the reaction time of student B. Student B then drinks a caffeinated drink and then they repeat the experiment 15 minutes later. Suggests one control variable they should have controlled. Well, there's lots here. And they tell us specifically don't refer to age or sex because they already mentioned that by saying 15-year-old girls. So we should control the caffeine intake of all the children. Okay, so they had the same caffeine intake. We've got the body mass, the same time of day they should have carried out the test, the same amount of sleep each student should have had, use the same hand each time. Did they all do it with their right hand? The students should have no previous experience, i.e., they haven't done the test before, and ingest the same volume and concentration of caffeine. Right, let's look at 5.2. Suggest 2 sources of random error when using this method to measure a student's reaction time. Well, student B may anticipate student A saying stop and press a little bit early. Student A may not start both stopwatches at the same time. Quite hard to click your thumbs down and start 2 things exactly at the same time. So that would lead to some error as well. When student A says stop, they might not press stop on their stopwatch at the same time as they say stop, which might mean there's an error in reaction time too. Okay, let's move on. 5.3. Why can a mode not be determined from this table? Have a look at this data. Why is there no mode in this data for the 8 pairs of students? Why is the decrease in reaction time after drinking a caffeinated drink in seconds not got a mode? And the answer is there isn't a multiple of any of the answers. There's not more than one of any result. So a mode is the most common occurring result, and obviously each result only occurred once. 5.4. The students decided the result from pair 8 was anomalous. The students calculated the mean. Look at this pair 8. They calculated a mean. Now explain how they then calculated their mean to get a reaction time of 0.030 seconds. Okay, so the correct method to do here when you spot an anomaly is to remove the anomaly, that's the 0.097 for pair 8, add up the remaining 7 results and divide by 7. Okay, 5.5. Caffeine causes the release of adrenaline. This is the adrenal gland here above one of your kidneys. Adrenaline affects the heart rate. Explain how the effect of adrenaline on the heart rate might cause reaction time to decrease, which basically means your reactions get faster. So adrenaline increases heart rate, which is going to increase the blood flow to the muscles. Okay, increases the blood flow to the brain, more oxygen to the brain, more oxygen supplied, okay, which means more aerobic respiration, which means you can release more energy, which allows you to contract your muscles faster, which gives you quicker reactions. So let me just go through and show you which of these answers appears on the mark scheme. So you've got increase in heart rate, increases the oxygen supply, more respiration, more energy for muscles, more contraction, uh, quicker reaction times. Right, let's zing into this next question. Adenosine is a different chemical made by the body. Adenosine binds to receptors on relay neurons. Adenosine decreases the number of impulse in relay neurons. This shows how caffeine binds to adenosine receptors on a relay neuron. When caffeine binds to the receptors, it blocks adenosine from joining. Label X, which shows the gap between the sensory and relay neuron. Well, the gap between any 2 neurons is known as the synapse. Suggests why reaction times decrease when caffeine binds to adenosine receptors. Well, if caffeine binds to the adenosine receptors, remember the adenosine reduces impulses. So the adenosine can't bind to the receptors as caffeine's in the way, blocking it. This means there's no reduction in impulses. So we get an increase in the speed of the impulses. Faster impulses mean faster reactions. Okay, let's move on. So we have 6.1. Some people are heterozygous for a genetic disorder. Define the term heterozygous. So that's when you have a dominant and a recessive allele. You have one of each allele in the genotype for a disorder. Example, big B, little b. Okay, let's look at 6.2. Figure 6 shows the inheritance of a genetic disorder in a family. So look, we've got 1 and 2 of her kids, 5 and 6, 3 and 4 of her kids, 8 and 9, 7 and 8 of her kids, 10 and 11. A white circle represents a female without the disorder, a white square is a male with the disorder, a black circle is a female with the disorder, and a black square is a male with the disorder. So that's how to interpret that. Figure 7, person 7 and person 8 have to have another child. Determine the probability that the child will be a male who has the disorder. You should draw a Punnett square. Identify the phenotype of persons 7 and 8. Okay, basically do a Punnett square of their next child and work out the probability that it's a male child with the disorder. So let's look at 7 and 8 here. We need to decode what they are. We'll look at 1 and 2. 1 and 2 produced 7 as their child. 1 and 2 did not have the disorder. Okay, so they didn't have that disorder, 1 and 2. What this means is that the disorder must be recessive. If you have two parents without a genetic disease and their child has it, it must be recessive. So they're big H, little h, 1 and 2. So number 7 must be little h, little h. This means 8 could be big H, or big H, little h. But 8 has already had a child 10 with the disorder. So 8 must also carry a recessive gene, a little h. How do we know that? Because their two little h's must have come together to produce 10. So now we know the genotypes of the parents. So 7, which is a male, is little h, and 8 is big H, little h. You just got to do the cross, circle and identify one of these phenotypes. So this is somebody with the disorder. That means there's a 50% probability they have a male or female child with the disorder. But the question asks specifically for a male child with the disorder. So just showing you here, the probability of you having a male child with the disorder is also 50% by doing our XY times XX. So we need to do 50% times 50%, 0.5 times 0.5. And the answer is 0.25. So there's a 25% chance of having a male with this disorder. You might want to pause and rewatch that. I'm going through quick. Okay, we've made it to the final question. Polydactyl is an inherited disorder. Two parents do not have any alleles for polydactyl in their ordinary body cells. These parents have produced a child with polydactyl. So how can they do that if they don't contain the allele for polydactyl in their body cells? Well, again, a random mutation in one of the egg or sperm cells of the parents could occur, and then this is passed on to the offspring. The mutation obviously causes a change in the DNA, and what does a change in the DNA do? Well, it changes the amino acid sequence of a protein, and that protein might cause polydactyl. So that essentially is how two parents without polydactyl could have a child with polydactyl. We can also decipher from this information that polydactyl must be dominant because they only need the random mutation to happen either in a sperm or an egg. So only from one parent, which means they are going to receive two alleles, one normal one and one that's not normal. Okay, that's that done. That was a bit of a rubbish walkthrough, but there we go, folks. Let's get
You're
to the
still
marks.
here? It's over.